The title of my presentation is Unlocking the Power of Fair Data Sharing with ImmPort. As Dr. Lee mentioned, that I'm a science program lead and outreach coordinator for ImmPort and bioinformatics project leader at University of California, San Francisco. So I will start with introducing the ImmPort team, the workforce behind ImmPort. On the far right-hand side is Peloton team. Peloton is contracted with NIDDK for curating public data sets from NIDDK researchers and harmonizing and curating the data sets. ICSF is our engineering team, software engineering team, maintaining databases, securing the data. And at UCSF, Dr. Atul Butte is the PI for ImmPort, and UCSF leads the scientific perspective or gives scientific perspective to ImmPort. We have Dr. Anupama Gururaj and Dr. Kwan Chen and Dawei, program officers for NIDDK. Definitely be getting the funding support from NIDDK. So it takes a village to build and maintain such large-scale data repositories. So today, the outline of my talk is I'll give a brief overview on ImmPort and then secondary data reuse with few case studies. So the molecular portraits of immune system with this with the recent trends of systems immunology, there's a lots of data getting generated in the proteomics, transcriptomics, genomics, and epigenomics area at the population level and at the single cell level. And so the question is, where do we store all of this data? So this is more on the clinical researchers asking the question that, we are good at generating the data, but then we need to keep our data safe, data well curated, and then also would like to share with the community. On the other hand, the clinical trial data, right? So there's lots of data getting generated on the clinical trial side, either individual participant data, summary data, metadata. Quite often we go to clinicaltrials.gov and look for data, and and there are various in this life cycle that lots of data getting generated in many different forms and shapes and sizes. And so those type of data could also be leveraged as a secondary use case. And so at that front, back in 2015, IOM had come up with sharing the clinical trial data, like kind of maximizing the benefits and minimizing the risks. And why don't we share all of this data so the secondary investigators can make use of this data to better design the clinical trials, forthcoming clinical trials. So, back in 2021, I wrote a perspective on opportunities and challenges in democratizing the clinical research data sets. As previously I mentioned that a lot of data sets are getting generated. There's a data trove, but at the same time, there's also computational tools, computational platforms are getting built to analyze such data sets. So data management, infrastructure, and computational resources, this all comes into play when we are talking about big data either in the private database, nonprofit, public repositories. And there are times when the data is siloed, it's sitting in someone's laptop. How do we bring this up and can be leveraged, right? So basically the question of accessibility, discoverability, interoperability. So today I'm gonna be highlighting one such repository, ImmPort, and I have been affiliated with ImmPort for last 12 years. And ImmPort came into existence about 20 years ago, and this year is our 20th anniversary. So ImmPort Data Portal was developed to collect and share research and clinical trials data from NIDDK or DATE-funded researchers, and we make our data fair, which is findable, accessible, interoperable, reusable. That's our mission. Within the ImmPort 6 ecosystem, we have private data space. So the investigators can store their data before it's been published, either in the form of kind of a publication and then also sharing the data through the publication. But before that, that stored data can be stored in the private data space. And once the researcher is ready to publish, then it gets we move it to the shared data space within ImmPort. And then we also have resources. So whatever we learn, we disseminate. So, resources could be tutorials, lots of documentations, and all the publications either coming out of ImmPort, those are available through the resources. And then data analysis. So we also develop tools to analyze our own data. And then we also release those data in the public domain for others to make use of. Those kind of, or take advantage of those tools. So the question is, who shares the data? Let's take a look. So ImmPort shares data from about 40 plus major NIDDK funded programs. And we also have external organizations submitting data to ImmPort. I will just highlight some of them. That we have data coming from HIPC, Human Immunology Project Consortium, Civics, Bill and Melinda Gates Foundation, ITN, AMP Consortium submitted data on lupus and rheumatoid arthritis. We have data coming from March of Dimes with maternal health. And we have about 265 studies as of today for COVID-19 from various other resources. One of the major one is SeroNet, submitting lots of data to ImmPort. So as you can see here, the list is quite long, and I won't be able to read all of them, but we are getting lots of data from different types of disease, and in a moment I'll show you what types of data we have. So we get this question that how do we submit data to ImmPort? So data submission process in ImmPort promotes fair data. So the major steps in data submission is first to create an ImmPort account, access a workspace. So a workspace is assigned to the data submitter. Then we register a new study, and then we complete, validate, and upload the data, which is more the data providers. They complete the templates that's been provided to them, and then they validate and upload the data. So we have two ways to go about data submission, but we highly encourage the data submitters to do a digital submission, so form-based online submissions. So we call it State Study Registration Wizard. So if you go to ImmPort website, there's a data upload on the navigation bar. You could just click and see the requirements. And it's a fairly a very easy process. There's few questions being asked, and then you can upload the data. And there's another route, which is like you can also make use of some data submission templates, which captures the metadata based on the study design. And we also keep a provision that if you have any new measurement techniques, which we don't support at the moment, but we also customize the templates, so we can parse the data into the database. So the submission templates also have control vocabulary terms. So in the process of submitting the data, you are also helping us because we provide you some control vocabulary terms. So basically to standardize and harmonize the data. Just a quick look at the data model. ImmPort is a relational database. We capture the data at a study level. Each study has subjects, and then subjects might be in groups or cohort or arms, depending on if it's clinical research data or trials. The terminologies change. So in a cohort, it could be a treatment arm and a control arm, and within each arms there are subjects, and then the subjects, the biosamples are collected from the subjects, and each node here is, if you follow from study, you have cohort and then the subject, and from subject you collect the biosample. For example, we collect blood from the subjects, and then for whole blood, and then probably for the downstream data capture, we run a bunch of experiments if depending on, like, you extract plasma or serum or any other biofluids, and then you run the experiments. And the beauty of ImmPort is that we support multimodal data. So we, as you can see here, a lot of immunology techniques here. We support ELISA, ELISpot, flow cytometry, and we are one of the largest flow cytometry repository, storing raw data files and as well as processed data files. We also store the protocol. So basically if you're looking for data transparency, how the data was generated, we also have associated protocols being curated for each study. And then we implement various ontology for data standardization. Here is some examples of, here are the table. Here is the table for study with attributes about the study. So it's pretty detailed and comprehensive in terms of data capture. In the interest of time, I'll start with the FAIR principles. First one is findable. So adherence to the FAIR principles, which increases the visibility of your data. So ImmPort has developed a cohort discovery tool. This is a tool which anyone can access. If you go to ImmPort website and you would like to look for a disease of your interest or study or data of your interest, you could do a free text search. We have a faceted search if you have very specific a query of interest, then you can look for or identify studies. And we also have associations with additional repositories, like for example, gene expression data. We take a federated approach. Like if you have a gene expression data, you have clinical data. Clinical data goes into ImmPort, and the gene expression raw data resides in Geo, but we do provide the references. So for that particular study, you can go to Geo and retrieve the gene expression data, or like that, many other repositories like dbGaP and others. We also you can access ImmPort datasets through the Vivli platform if you're interested in the clinical trials data, which are in ImmPort. So here is this ImmPort shared data browser, which we call cohort discovery tool. So ImmPort currently shares over 900 studies, which encompasses a range of research areas, different species. We are big on humans, and then mouse, and then we have other species as well. And then there are over 25 data types that we support, and about 188 clinical trials as of today are getting shared through ImmPort. And here is the QR code. You can just scan and go through the website. Here's a link also provided if you're interested to look for the disease of interest. And so just to give you a data summary, so we do a monthly data release. So back in March, here are the numbers. We released 927 studies. These are incremental, not that we released 900, but in the last data release, we reached to the point of 927 studies. And as you can see on the pie chart on your right, as this is the distribution of type of studies. We have lots of vaccine response studies, immune response, infection response, transplantation. We are big on transplantation, allergy, asthma, allergy, and also autoimmune diseases. So we have lots of vaccine studies that we have a lot of researchers have leveraged in their research studies. And I'll come to that a little bit later. And if you look at the table here, the numbers for ELISA, ELISpot, and many different measurement techniques. We have also, under flow cytometry, we have CyTOF data. We have a lot of Luminex data for cytokine profiling, and neutralizing antibody results, and also lab test. So we have clinical trial data, and we have lab test data. Clinical trials is not covered here, but we have lots of lab test data with many different metabolic panels and CBC blood panels and many others. I just pulled out something probably might be of interest to this group is if you're interested for any kidney or liver or diabetes, then based on the condition using the CDG tool, I looked at how many of the studies are available. And so you can see here that we have about 41 studies on kidney transplantation, and then about 3 on liver transplantation and lung transplantation, heart transplantation, and then type 1 diabetes, kidney failures, chronic kidney disease, and so on. Now let's come to accessible. How do we access these data sets, right? So that question gets answered through registering to the ImmPort website. So registration to ImmPort is free. What we need is just your email address and affiliation, and you're ready to go. So once you agree to the terms, which is just, be mindful about when you download the data and make a good use of the data. So you just sign up for that agreement, and then you are ready to download the data. Registration is free. And then there are many ways that the data can be accessible. It can be that once you go to the web page, identify the study, you can download in a tab delimited format, SQL format, and also we have API. So ImmPort provides APIs to programmatically download the data, depending on if you are interested in for example, lots of flow cytometric files, and these are raw files, large files. So through the API, you can call and download all of the data on your local computer once you have registered and agreed to the terms and conditions. In terms of search, there is no registration required. You can just go and search on the ImmPort webpage. Now to interoperability. So we are trying to make our resource interoperable with other resources. There's a big initiative going on for FHIR, which is a fast healthcare interoperability resources format. So we are trying to standardize our data sets in a FHIR format so that we can crosswalk towards through the different repositories, either Geo, dbGaP, or other repositories, those who support FHIR format. And we have currently a project going on where we are trying to access data sets which resides in dbGaP, Geo, and ImmPort. How can we, through a single interface, call the data which are in 3 different repositories from a single interface or from one interface, rather than going to 3 different repositories, which is time consuming, and also it's difficult to find where the data sets are, how to map the subjects across the repositories from the same study. We do have ImmPort FHIR server, for which we have some of the data sets which are available through the FHIR format. And in the future, we're going to be providing FHIR format for all the studies in ImmPort. So far we were talking about how to generate the data, how to curate the data, how to submit the data to the database. But how about what are the benefits of open access immunological data? So my team basically focuses on repurposing open access immunology data sets. In this case, is ImmPort. So basically, to facilitate the reproducibility, secondary analysis of the data sets and repurposing. So, oops. So here is the cycle that typically a research or investigator, they generate a hypothesis, run an experiment, and then they generate data and then ask a question. There's a model. And so we come in as a data scientist. Ours is a data-driven hypothesis. We log into repositories, in this case, ImmPort, look for studies. For example, if my interest is in autoimmune disease, I'll look for all the autoimmune disease, try to download the data and see what questions can be asked, which was not formulated by the primary investigators. So here is such example. This is one of my favorite slides from ImmPort, where we were trying to identify influenza vaccination cohorts. And as you can see here, it's a crowdsourcing approach, because the data is coming from different sources, right? So that's the beauty. So we can holistically look at this data where the common theme is all these individuals are vaccinated but these are different cohorts. There's a healthy cohort. This big bubble is healthy cohort, vaccinated, 843 individuals. Then comes here is this cohort, lupus cohort. They are all vaccinated, but have lupus. So that is, or probably aging, vaccinated cohort. And then we are starting to study the effect of age on flu vaccine. Or here, if you are doing some genetic studies, we have data on twins who are vaccinated, atopic dermatitis, and many others. So this is only possible when data is coming from different sources. And then as a data scientist, we go in and then query the data and then ask questions. So this is a good use case of how so many questions can be asked because you have this data made available through the primary investigators. So we are very thankful to the data providers because this kind of research can be done since the data is getting deposited in a central location. So I will just highlight a few of the studies that my group did. So this was back in 2018. We made a big splash where we developed a 10,000 immunome project. So basically the idea was that in the genomics world, there's a 1,000 genome project, which is a reference. But in immunology, there's nothing like that existed. So from the ImmPort studies, we just looked at the normal healthy individuals. So we kind of subsetted the data, looking at the control arm or healthy individuals or healthy subjects. And then we subsetted the data to build a large, diverse, clean reference data set for human immunology, and then build an interactive data visualizations and provide custom control cohorts and standardized data downloads. So let's spend some time here. So basically, we started with 85 studies within ImmPort with 10,000 subjects, for which we had cyto for flow cytometry data, some of the lab test data, gene expression data, Illuminex data for cytokine profiling, and so on. So here is the list of the measurement types. As you can see here, in clinical labs, CBCs, metabolic panel. We also had questionnaire, cytometry data, HLA typing, gene expression array for whole blood and PBMCs. And so we built a standardized pipeline for data cleaning and harmonizations. And once this data was standardized, we disseminated data so that the community can also leverage this kind of standardized, harmonized data for their own research. Here is the interface. So if you log into 10K Immunomes, again, there's no it's not actually, there's no registration or login. You can just go onto the website, 10K Immunomes, and then start looking at if you have any particular data types of interest. You can look at what is the gene expression in normal healthy individuals, or cytokine concentrations in normal healthy individuals, or cell types. So it's pretty comprehensive that we did transcriptomics, proteomics. Each of them are tabs, and you can go to each tab and then also look at the data. In not only just you can visualize the data depending on what your interest is, and on the fly you can select the age range or which study it's coming from, and then if you have interest in race or any other social determinants of health factors, you can see it's right now limited to demographics, but we plan to expand it. But you can either take away the figure, or you can also download the underlying data. So from last year, we had been also thinking about how can we make all these data set AI ready. So 10K Immunomes is a pretty standardized data format we had. So this is just an example that you can download the data from 10K Immunomes and plug into ChatGPT, ask the questions as what is the effect of age and race. The one I showed previously was just for one particular case where you're looking at the effect of race, but how about you large scale, you can download the data, and through the ChatGPT using some prompts, you can study and ask questions. What are the age associated difference in cell types, or effect of cytokines on age and gender, and so on. So this holds a lot of promise in terms of, gives you lots of promise on making these data sets AI ready. So ImmPort very soon is coming. where we will be releasing the AI-ready data sets. These are import data sets. We'll make it AI-ready. The first one is going to go out as the 10K genomes, and then also the Ko-Wei data sets will be the AI-ready data sets that you can start analyzing right away, because we'll be providing the standardized data. We also did a kind of a nice project on a deep learning model for cytometry data. So we used a convolutional neural network for cytometry data to diagnose the latent CMV, cytomegalovirus in healthy individuals. So using the deep learning model, can you predict that, since it's latent, like who are CMV infected and who are not, based on cell types? So in the interest of time, I won't be able to go through the project in detail, but we got a pretty good success on identifying the cell types, which can predict like who are CMV or non-CMV individuals. The paper came out in PNS in 2020. And then there's another one, which was an ambitious project based on a study. We have lots of transplants data in ImmPort. So this is back in 2018 when we had 27 clinical trials on organ transplant. And so as you can see here, the pipeline that we had 27 clinical trials, we curated 20 from about 11,000 subjects. And so we compiled the data, standardized the data, and the data had demographics. We had pre-transplant data, intraoperative post-transplant, and the recipient data as well. But we were focusing on donors. And so we were looking at, as here's this trajectory analysis, that after donating the organ, the donors were followed for max 40 years. So if you can see here that anytime from 2 to 40 years, and we were seeing what kind of complications arise if there were, and you can see here some of the complications arise after donating the organ. And so all the data was available in ImmPort in the form of survey questionnaires, and the data made available, which helped us to build this kind of trajectory analysis. And as you can see here, some of them like hypertension, and then some of the other secondary complications showed up. So this is all based on data. So, we were just looking at the post-donation outcomes here. So this was one of another use of data coming from different CTOT studies. And as you can see here that we have about 20 clinical trials data from RELIEF, and then we were able to build this trajectory. So like this, over the years, we have a lot of publications of data you use, big data, meta-analysis, we build meta-analysis pipeline, machine learning. So today I highlighted a few of them, and then we also wrote a lot of reviews. And so as you can see here that there's a lot of opportunities from this open access data sets. So previously I showed you all that we have done, but there are also researchers outside of ImmPort team, those who have leveraged ImmPort data. And when we were into pandemic, there's a group from Stanford, from Bali Pulinjan's lab, and they looked at some of the data sets, those from ImmPort. So as you can see here that we have data sets for flu, varicella, VZV, yellow fever, Ebola, HIV. And when they were looking at the BioNTech vaccine, there were no other reference, right, for them. But for them, the reference was all that listed here, which are like from other vaccine. That came in handy because all this data was available in ImmPort. And so the researchers, they downloaded the data and then compared the BioNTech mRNA vaccine to the vaccines from other pathogens. And so this is just to give you an idea that if the data is well curated in a pandemic like this, which comes without any notice, right? It's just, then these kind of resources are very, very helpful as a reference for the researchers to compare their data sets. Lately, there's another data use case coming from Sandra Endorf. She's the senior author on this paper, and we have a LEAP trial, which is the learning early about peanut allergy data set in ImmPort, and they also asked questions because lots of data, LEAP trial data is in ImmPort and through the ITN trial share, and they leveraged the data to ask, again, a question which was not formulated before, so to get insight into peanut allergy, or who are the risk subgroups and intervention effects and so on. Also, Immune Space, part of the Human Immunology Project Consortium, they also get the data from ImmPort, and so HIPSI's Immune Space gets access to the data. They standardize the gene expression data, and if you go to the Immune Space website, they basically try to understand the data, kind of a high-level analysis. ImmPort doesn't analyze the data. They provide you the data sets, but then here are the groups, those who download all of the ImmPort data, and then they study either the flu vaccine data sets or other data sets and make the data and turn it into knowledge, basically, and make it available to the community, to the research community. So if you have interest, take a look at immunespace.org. This is primarily focused on the flu or vaccine cohorts, but could be of interest. Education. So, what I previously mentioned, whatever we learn, we disseminate. So when we analyze these data sets, we turn it into tutorial. So if you are new into this space, or graduate students, researchers who would like to touch the data or analyze the data, they might be generating lots of data, but they don't know how to analyze data. So this we have provided tutorials where we step by step, or we showcase how to download the data and then look at the metadata files, combine the data sets, and then ask questions. So follow the tutorial on ImmPort page for learning. So the take-home message, what I've shown so far, is that open access immunological studies are a valuable resource to evaluate new in silico hypothesis testing, gain novel insights, and a productive starting point for informing the design of future experiments. The vaccination data that I showed you with bubble charts, like that's that holistic approach in analyzing clinical research data. 10,000 Immuno projects, which is a framework for growing a diverse human immunology reference for ImmPort or publicly available resource at the subject level, not just summarized data, but subject level data, which allows you to learn from the features and the candidates. You can run, deep learning or AI models on the standardized data sets, and which enables us to explore new factors to be discovered. We did a CNN deep convolutional neural network model. We developed a model to accurately diagnose the latent CMV in healthy individuals. It was an ambitious project. We didn't know what to do, what will be the outcome, but we were great when we found that we were able to identify based on data sets. These are all big data, which allows you to run this kind of models. Expanded uses of crowdsourcing in immunology will allow for more efficient large-scale data collection and analysis. It will also involve, inspire, educate, and engage the community in a variety of meaningful ways. I hope I was able to convince you to embrace open access datasets. If you want to stay updated on ImmPort activities, we release monthly newsletters, which comprises of the news events, the webinars that we conduct, and then new data releases. All of that informations are available through the monthly newsletter. So if you're interested, please subscribe and stay connected to ImmPort. We are big on social media. We also announce data releases on social media. And then if you want to reach out to us on one-on-one basis, we have office hours, ImmPort help desk. If you have any questions, you have any disease or conditions that you want to know if the data sets are available, or can I submit data to ImmPort? Those kind of questions can also be asked through the help desk. So ImmPort office hours, we hold open office hours session on first Thursday of each month. And it's a great opportunity. If you don't want to ask questions in a public forum, you can also attend the office hours and discuss the questions with the ImmPort team. And all user levels are welcome. If you are a bench scientist, if you are a data scientist, if you're a citizen scientist who wants to know more about the data sets or create awareness, you are most welcome. And here is the link for the office hour times, and you can register to, I think it's a Zoom meeting, so it's pretty interactive. So this June, I'm offering a workshop on big data in immunology. For the last 6 years, we had been conducting the workshop at Focus, which is the Federation of Clinical Immunology Society. This year is in San Francisco. We have a panel of speakers. So we have, this workshop has 2 parts. In the first part of the session, one is about, we invite speakers, those who have leverage ImmPort data or generating big data. And then the second half is hands-on, where ImmPort team goes in, drills down to the data sets, few data sets, some example data sets, and then run an exercise on how to retrieve the data from ImmPort, and then using either R or Python notebooks, how to analyze the data. And this is open for all. Anyone can join this workshop and then learn about the ImmPort data sets and the types of research that's happening around ImmPort data sets and projects and initiatives that we had been kind of pursuing for last so many years. So with that, I would like to thank everyone for your attention, and happy to take questions.
Okay, thank you so much for the great presentation. And now is our Q&A session. So you are welcome to unmute yourself to ask question, or use chat function to just type the question there, and I will ask the presenter. And we are also welcome, I think Kate Hypes will also join us during this session. Yeah. Yeah, so I saw a question from the chat during your presentation. The question is, I think it's probably coming from the first 10 or 20 minutes presentation, so maybe we already covered that in the presentation. The question is, does the data need to be in a clinical trial to be deposited in the ImmPort, or it's only restrict to the human subject data, research data?
No, there is no restriction. It could be clinical trials data, it could be clinical research data, but we make sure that it's everything in consented and de-identified. So we also help in the de-identification process, but we want to make sure that, yeah, probably I forgot to mention that these are all de-identified data, subject level de-identified data.
Okay, thank you. And the second question I got from the chat is, okay, let me repeat that question first, and then I will ask the question from Jing Yuan. Okay, so the questions I got from the audience is, do you have any suggestion when I download the data from ImmPort and use it, how to cite it?
That's a very good question. So if you have, or what the researchers have done in other publications, when they download the data, each ImmPort study has a study accession number. So in the data available, when you are downloading the data, then you can say the data was downloaded from ImmPort. Here is this SDY number. So each study has accession. And then depending on if it's a secondary data reuse, we also cite the investigators who have submitted the data. So because they are the ones who generated the data. And then when you're downloading, your downloading source is ImmPort. That's why you mention ImmPort, that you download it from the ImmPort repository, and here is the study number. So I would say that we should acknowledge the group, those who have submitted or provided the data, and then the repository, and then whatever you're doing with the data. If you're using it for secondary analysis, or you're combining your data with the study data from ImmPort, it depends what the purpose is.
And Sanchita, I will also add to that too. If you go to the ImmPort website, up in the upper right corner, there's an About drop-down menu, and it actually, you can find a page there that will give you some examples of exactly how to cite, types of data sets within ImmPort. So very good question. Thank you for that. And I'll say too, it's also part of the data use agreement with ImmPort that we encourage those who use the data to please acknowledge and cite the providers, whenever possible.
Okay, thank you. Hope that answer your question. If you have more questions, just type there and I will repeat it. So there's a question from Ji Yuan. Is the data free of use? Should we submit a proposal or collaborate with ImmPort research to be able to get access to analyze data? Thank you.
If it's data reuse, you just need to register to ImmPort, provide your email address, and you are ready to download the data. So, but if you are asking that how can I submit data, probably if you are a NIDDK investigator, it's free to use. We will provide you the service to upload the data and make the data available in a shared space. If you are an external investigator who is not associated with NIDDK, you are welcome to write to us, and then we will consider if these are relevant data sets for ImmPort, then definitely this will go under review and we go from there.
Yeah, and I will add to, while ImmPort, like we said, is funded under NIDDK, we do have a number of data sets. So as long as the data set is appropriate for the type of data that ImmPort collects, we know there's a lot of crossover amongst many centers, with immunology. So basically, if it's immunology related data and within data types that ImmPort selects, we're very happy to receive it. And like Sanchita said, if you contact the ImmPort team, then we can kind of understand your data a little bit and advise you whether it would be a good fit or not for ImmPort. And I will say too, that does segue a little bit into, I think, top of our minds recently is the data management and sharing policy from the NIH. I know a lot of folks are looking to find a good or correct repository to share the data with. I will mention ImmPort does have resources for that. So if you go again out to the ImmPort page, up at the very top bar, you will see data management and sharing plan, and you can click on that there, and it will give you some insight into what data types ImmPort accepts, some tools and templates and things to help you with data management and sharing plan creation, and then of course our information that you can reach out and discuss with us further.
Okay, and then I see in the chat there is a link shared by Liz. I think that's about the is that how to cite? Yeah, just let everybody know. It's the docs.immPort.org/home and /site for those who couldn't see the screen. Okay, and then there is a question from Dr. Martin. Why isn't ImmPort adhering to the recommendation of the data citation principles so that citation can be automatically tracked?
So Yes, it's great question. And so there is an effort underway right now with DataCite and some others to do that type of automated tracking with citations. It's kind of developing and ongoing, and ImmPort certainly does, hope to become involved with that more in the future. So yeah, appreciate that question, because I think it's very, very important that we can, acknowledge and cite as best as possible everywhere. Yep.
Okay.
I would like to add one more thing here, that for each of our data sets, we have a DOI. So if you want to use the DOI, that's another way to go. So our data sets, all of them have DOIs.
So I would recommend you update your citation guidelines now, because yes, it is true what DataCite is trying to do, because it's been very difficult to get all the publishers on board to actually extract the citations. But if you do recommend that they put that DOI in, that's what they're going to go for. And also put it into the reference list, which is the main recommendation. But what we often find is people say, I got it from ImmPort. They may put a number, but really I think it's the DOI that you want them to put into the paper, and even the data availability statement, so that those can be extracted and tracked.
Yeah, great suggestion, and I think we'll follow up on that.
So anyone from the participant want to unmute yourself or ask question or have some comment to discuss?
Hey, Ko-Wei, this is Chen from UCI. We have some questions.
Sure.
Can people hear us? Yeah, so hi, Sanchita, very nice work. This is our team from UC Irvine. You can see we have quite a few students here besides me, because the work is very interesting to us. And thanks for the presentation. I have a general question. I see there are a lot of users of your website, the data sets, and they have done a lot of analysis. And your team also did a lot of analysis. Based on your interactions with your users, do you see the need for your team to share some of the analysis with all the other users, or also for the users to share some of their analysis with other community users?
Yeah, so I can speak for our analysis. We have made it available through the tutorials. And so if you go to our website, you'll see either on the website or through GitHub. So if you read our papers, one of the ones that we did was a Metacyto flow cytometry pipeline. If you go to Metacyto paper that we published in Cell Reports, we have a GitHub link. So all of the codes are available. For 10K mutants, how did we build the resource? We have a Docker image for 10,000 genomes. We have a GitHub page which have all the codes. How do we standardize the data and make the resource, right? Because this resource was built on our Shiny application and we made it interactive. How did we standardize the data? So basically, we have GitHub links. So that's from our team, whatever resources we built. And some of them are also in the form of tutorials on ImmPort website. Now, you raised another very nice point here. Good point is that, or interesting, I would say that, how about others who have? For them, I have no control, but we can ask them that if they can share. The BioNTech was one of them, which the example I showed you from Stanford. I'm sure they have the codes, how they downloaded the data, and how they analyzed the data, which generated that figure comparing the BioNTech vaccine with others. Maybe going forward, we can put a request that if you are reusing ImmPort data and coming out with a publication, will you be willing to share the code that the community can make using it? Yeah, very, very, I would say we never thought about it. It's an interesting point that we should consider. Yeah, as a giveaway or education. Yep.
Okay. Does that answer your question, Dr. Lee?
Yeah, I mean, one reason I asked the question is 2 weeks later it's going to be my turn to give a talk. so I'm just using this chance to to mention some of the ideas we have been pursuing, which is exactly on this topic, how to allow the community users to easily share their analysis in addition to sharing data. And data is very powerful, very valuable. We believe there's a significant value also in their analysis. Some code, some pipelines, and it'd be nice if we can have a framework or infrastructure that can allow people to also easily share their analysis. So that's why this is something our team is very interested in, and some other dkNET colleagues, some of them are also on this call. We are working together toward that goal. So that's something I would like to talk to you more about. If something.
There are discussions that happened within ImmPort team with also NIDDK that how I would make the code available, right? Code availability. Now, some of the journals are already asking that, when you did this bioinformatic analysis, make the code available. I have seen papers where they have made the code available. Not all of them, but there are. And I think it's a good practice. If the journals can also make it as a requirement, the way they have made the requirement that you need to share your data, the underlying data, right? So if they also make it as a requirement that make the codes available, that will be super helpful.
Okay, yeah, more can be discussed later. I just want to bring up this issue. If you're interested in that direction, we can talk offline about some of the work we are doing and see if there's any like a collaboration opportunity here.
Sure. Happy to.
Thank you.
Okay, are there any more questions from the audience? I'd like to unmute. I think there is a quick question from the chat. It's about the workshop that you mentioned in June. Do I have to be the data owner to attend the workshop? What is the requirement skill for attending that workshop? I think it's while you present, it's a big data in immunology workshop. Yeah, the question is coming from that.
Yeah. No. We don't require at least no programmatic skills, right? Because we expect, we welcome everyone. Because the workshop is meant for, we would like to give you all these, the type of work we do and how you can learn from that, right? And so it could be anyone can join the workshop. Now, in terms of attending the conference, it depends. We are part of the conference. You don't have to, I don't think you have to register to the conference to attend the workshop, but there might be a minimal fee to attend the workshop, which is managed by Focus. But from our side, there is no requirement. Everyone is welcomed.